Good morning, dear students. Today we can study exotoxins. Microbial toxins, they are poisonous substances produced by microorganisms that alter the normal metabolism of host cells and thereby causing deleterious effect on the host. Toxins are either protein, peptide or steroids. Toxemia refers to a condition caused by the toxin that have entered the blood of the host. Poisonous effect of the toxin may be due to the membrane damaging effect, cell surface activity such as interferences with the signaling and transportation processes or causing intracellular antagonistic effect. Toxins produced by microorganism is divided into two main categories, namely endotoxin and exotoxin. Toxins are poisonous substances produced by microorganisms which cause deleterious effect on the host cell, and toxins may be protein, peptide or steroids. And the toxemia, it refers to the presence of toxin or effects of toxin in the blood of the host. Then poisonous effect of the toxin may be either due to the membrane damaging effect of the host cell due to the effect of the toxin or due to the interferences with the normal signaling and transportation pathway of the host cell or many other antagonistic effect that are caused by the toxin inside the host cell. Then toxins, they are mainly divided into exotoxins and endotoxins. Exotoxins, they are proteins, mostly enzymes, which are produced by gram-positive and gram-negative bacteria. They are secreted out to the external environment or diffuses out through the membrane. Their molecular weight ranges from 50 to 1000 kilodalton, and specificity is very high, and they are heat labile, extremely poisonous with a potency of creating lethal conditions as low as 1 microgram in humans. Botulism, diphtheria, tetanus are diseases caused by exotoxins. Exotoxins, they are proteins, and most of the exotoxins are enzymes. They are produced by gram-positive. Most of the exotoxins are produced by gram-positive bacteria. Some exotoxins are also produced by gram-negative bacteria, and they are secreted. They are called exotoxins because they are secreted out into the external environment from the bacterial cell, or they diffuses through the plasma membrane of the bacterial cell, and their molecular weight is from 50 to 1000 kilodalton, and their specificity is very high, but they are very sensitive to heat. They are extremely poisonous. Even 1 microgram is enough to kill a human, and a lot of diseases are caused due to the effect of exotoxins. Botulism, diphtheria, tetanus, cholera, etc. are some of the diseases that are caused by exotoxins. Classification of exotoxins. Exotoxins are classified into different categories based on the site of damage that they cause and the kind of cells that are affected. Neurotoxins. Neurotoxins are toxins which affect the central nervous system. Example, botulinum. Then enterotoxins, they affect the intestinal cells. Example, cholera toxins produced from Vibrio cholerae. Cytotoxins, they affect different kinds of cells. Example, diphtheria toxin. Cytotoxins are again classified into leucocidins, which affect the WBC or leukocytes, and hemolysins, which affect or which cause the lysis of RBC. So, classification of exotoxins are based on the site of damage that the exotoxins cause or the kind of cells that they affect. So, neurotoxin, central nervous system. Neurotoxin affects central nervous system. Enterotoxin affect intestine. Cytotoxin affect different kinds of cell. Leucocidins affect leukocytes, and hemolysin affect RBC. Features of exotoxins. Exotoxin lost their toxicity when treated with formaldehyde, and then they are called toxoids. Their antigenic properties can be retained and have the ability to stimulate the production of antitoxins. Antitoxins are antibodies that react with the toxins and neutralize them in the body of the host. Toxoids are widely used as vaccines for immunization against tetanus, diphtheria, toxin, etc. So, exotoxins, they can be made inactive by treating with formaldehyde. They lost their toxicity by treating with formaldehyde, then they are called toxoids. Though they lost their toxicity, they retain their antigenic property. That means they can stimulate the production of antitoxins against this toxins. So these antitoxins are actually antibodies that are produced by a host in response to these toxins. So toxoids are enough to for the creation or the production of antitoxins. And this antitoxin help to neutralize the toxin within the body of the host. Toxoids are widely used for the preparation of vaccines since they are attenuated toxins. So they never cause toxicity to host cell, but they can elicit an immune response in the host cell. And examples of using toxoids in immunization are against tetanus and diphtheria. So tetanus toxin and diphtheria toxins, their toxoids are used in vaccination. Then general mechanism of action of toxins. Adherence of exotoxins to the tissue is an important step in their toxic activity. Attachment is usually made by a particular region or subunit of the toxin. Other subunits of the toxin may have enzyme activity that causes damage to the cell. Some exotoxins act directly at the ribosome to inhibit protein synthesis. An example is Shiga toxin. So, in order for a toxin to act, it must first attach to the surface of the host cell or the tissue. So, adherence of the exotoxin to the plasma membrane of the host cell is the first important step, and it is usually attachment is usually made by either one of the subunit or the one of a particular region of the toxin. So, toxins are toxins are made up of complex subunits. So one or two subunits will help into the attachment, and other subunits, they have the enzymatic activity which is responsible for causing the actual effects of toxin. So other subunits of the toxin, they have enzyme activity that cause damages to the tissue cell. Some exotoxins, they act directly on the ribosome, inhibiting the protein synthesis. An example of such toxin is Shiga toxin. You have to study 4 main toxins. First one is diphtheria toxin. Diphtheria toxin is secreted by Corynebacterium diphtheriae. Diphtheria toxin is a single polypeptide chain containing two major domains, A domain and B domain. A domain has enzymatic activity and is cleaved from the B domain before entering the cell. B domain, which promote binding of the toxin to the cells and promote the entry of A chain into the cytosolic compartment. Diphtheria, it is caused by Corynebacterium diphtheriae, and the main cause of diphtheria is the production of diphtheria toxin by Corynebacterium. So diphtheria toxin, it is a single polypeptide. It is composed of two domains, A domain and B domain. So A domain, it is having the enzymatic activity, and A domain and B domain is attached as a single polypeptide, but since A domain is having the enzymatic activity, it has to enter into the cytosolic compartment to get activated. B domain help to attach the whole diphtheria toxin into the plasma membrane of the cell. So B domain, it helps in binding of the toxin to the cell. Once the B domain or the toxin get binded to the plasma membrane of the host cell, this A domain get cleaved from the B domain and it will enters into the cytosolic compartment. Diphtheria toxin kill cells by inhibiting eukaryotic protein synthesis. This potent toxin inactivate elongation factor 2 required for protein synthesis. A domain carries the active site for ADP ribosylation of the elongation factor 2. It catalyzes reactions that inactivate elongation factor EF2, essential for the elongation of the growing polypeptide chain. It results in the inability of the tissue to no longer make proteins and cell eventually dies. So, when diphtheria toxin enters into a eukaryotic host cell, it inhibits the protein synthesis there. So, here it specially target into the elongation factor 2 that is responsible for the elongation of the growing polypeptide chain in the ribosome. So, elongation factor 2 is inhibited by the A domain of the diphtheria toxin. A domain, it carry the active site for ADP ribosylation of EF2. So, EF2 it is usually seen attached with a GTP. ATP. So, EF2 ATP it is being deactivated to EF2 ADP by the A domain. So, this EF2 get inactivated and it cannot perform the elongation of the polypeptide. So, it results in the inability of the cells to no longer perform protein synthesis and eventually the cell dies. So, this is the action of diphtheria toxin. So, diphtheria toxin it is a single polypeptide chain composed of a domain and a B domain. A domain it helps to B domain it helps to bind the toxin to the plasma membrane of the host cell, and B domain then cleaves from the A domain, and B domain promotes the entry of the A domain into the cytoplasmic compartment of the host cell. And this A domain it is having the enzymatic activity and it is having the ADP ribosylation activity of the elongation factor 2, which is responsible for the elongation of the polypeptide in the eukaryotic cell. So, EF2 get deactivated by the ADP ribosylation by the A domain of the diphtheria toxin. So, it cannot perform the elongation of the protein, and the cell itself as a whole cannot perform protein synthesis no longer, and it eventually dies. Antitoxins. Antitoxins react with the B portion of the toxin, prevent the toxin from attached to the tissue cell. So, antitoxins it has the capacity to react with the B part of the or B domain of the toxin. Thus, B toxin or B domain cannot able to bind to the plasma membrane of the tissue cells. If attachment of the toxin into the host cell has already occurred, then these antitoxins cannot act on the toxin. So, prompt administration of the antitoxin is required in case of diphtheria. So, before the production of these toxins, the antitoxin has to be administered in case of diphtheria. Second toxin is tetanus toxin. Tetanus toxin is an extremely potent neurotoxin produced by the vegetative cells of Clostridium tetani in anaerobic condition. It results in tetanus. Tetanus is caused by Clostridium tetani, and the tetanus toxin is produced in anaerobic condition and it will result in tetanus. This tetanus toxin is otherwise known as spasmogenic toxin, but because it causes spasms, and it is also known as TENT, that is tetanus neurotoxin. Mechanism of action of tetanus toxin. Tetanus toxin initially binds at the presynaptic terminal of the neuromuscular junction. It is then transported by the motor neuron into the spinal cord. So, at first, tetanus toxin it will bind in the presynaptic terminal of the neuromuscular junction, and from that presynaptic terminal of the neuromuscular junction, it is transported by the motor neuron into the spinal cord. In the spinal cord, tetanus toxin is then transferred to the inhibitory presynaptic terminal surrounding the motor neuron. So, in the spinal cord surrounding the motor neurons there are inhibitory presynaptic neurons. So, it will This tetanus toxin then bind to this inhibitory presynaptic terminal. So under normal condition, this inhibitory presynaptic terminals will secrete inhibitory neurotransmitters which will render the motor neuron temporarily insensitive to stimulations through amino acid glycine. So under normal conditions, when a muscle is contracting, then the inhibitory neurotransmitters will cause the temporary insensitive, it will temporarily insensitive the stimulation through the amino acid glycine, so that the muscle contraction will be relaxed. So by the combined action of these two neurons, the muscle contraction and relaxation will be occurring simultaneously. But when the tetanus toxin get attached to these inhibitory neurotransmitters, then the toxin destroy a vesicular synaptic membrane protein, that is VAMB, and resulting in the inactivation of the inhibitory neurotransmission that normally suppresses motor neuron and muscle activity. So when the tetanus toxin is present, it will bind with the vesicular synaptic membrane protein which is present in this inhibitory neurotransmitters, and it will result in the inactivation of these inhibitory neurotransmitters, which will in normal case will suppress the motor neuron activity so that the muscle contraction is being stopped and it can relax in the next second. The toxin inhibit the release of the glycine. So this inhibitory neurotransmitters cause the temporary stoppage of the stimulation of motor neurons through the amino acid glycine. So the toxin, it inhibits the release of glycine, and as a result the opposing set of muscles often contract simultaneously and is in an uncontrollable fashion. So the muscle always get contracted simultaneously and in an uncontrollable fashion. So it will result in an enhanced excitability of the muscle and activation of the affected motor neurons. So motor neurons get activated all the times, and it will result in enhanced excitability. So muscle spasms will occur. So this is the mechanism of action of tetanus toxin. So tetanus toxin, it will bind with the neurotransmitters present in the inhibitory neurotransmitters that are present in the motor neurons in the spinal matter, present nearer to the motor neurons in the spinal cord, and it will affect their vesicular synaptic membrane protein, which will result in the inactivation of these inhibitory neurotransmitters, and it suppresses motor neuron, and it causes the regular excitability of the muscles, and muscle contraction will be occurring in a irregular fashion or an uncontrollable fashion. So muscle always get excited, and this motor neurons is always in a activated state. So that is the effect of tetanus toxin. Cholera toxin. Cholera is caused by Vibrio cholerae. Cholera toxin is an oligomeric complex made up of 6 protein subunits. It is composed of an enzymatic A subunit and pentameric B subunits. Subunit A activate the G protein, which activate adenylate cyclase. Cholera toxin. Cholera is caused by Vibrio cholerae, and Vibrio cholerae produces cholera toxin. Cholera toxin is an oligomeric complex composed of 6 protein subunits. 5 of these subunits are formed of B domain and a single A domain or A subunit, which is enzymatic in nature, and this subunit A, it activate the G protein, which in turn activate adenylate cyclase. Mechanism of action of cholera toxin. B subunits are responsible for binding of the toxin to the surface of the epithelial cells known as enterocytes of the intestine. B subunit of the toxin interact with the GM1 ganglioside receptor on the intestinal cell, triggering endocytosis of the toxin. A subunit penetrates the cell membrane and is cleaved to form A1. The enzymatic A1 fragment of the toxin A subunit enters the cytosol where it activates the G protein through an ADP ribosylation reaction. This locks the G protein in its GTP bound form, thereby continually stimulating adenylate cyclase to produce cyclic AMP. The high cyclic AMP levels activate cystic fibrosis transmembrane conductance regulator. It results in drastic loss of ions and water from infected enterocytosis into the lumen of the intestine, leading to watery diarrhea. 10 to 12 liters of fluid is excreted per day and the pH of the blood falls. So, mechanism of action of cholera toxin. The B subunit, it is responsible for binding the toxin to the intestinal cells. So, intestinal cells are called enterocytes. So, B subunit, it will interact with the receptor on the intestinal cell. Receptor is GM ganglioside. So, the B subunit interact with the GM ganglioside, GM1, not GM, GM1 ganglioside that is present on the intestinal wall, and that binding triggers endocytosis of the toxin. That means the toxin get into the intestinal cell. So the endocytosis of the toxin will take place when the B subunit bind with the receptor. So only the A subunit will penetrate into the cell membrane, and when the A subunit penetrate into the cell membrane or into the intestinal cell, it is cleaved to form an A1 fragment. So A1 is the activated form of the A subunit. So it is cleaved to form the A1 fragment. The enzymatic A1 fragment of the toxin A subunit enters into the cytosol, where it activates the G protein. So A subunit is cleaved to form the A1 fragment, which enters into the cytosol, and it activates G protein through ADP ribosylation reaction. So what happens is that the G protein will get locked in the GTP bound form. So GTP bound form is the activated form. So G protein will be found as G protein bounded with the GTP. So it is the activated form, and this G protein bound to GDP will continually stimulate adenylate cyclase to produce cyclic AMP. So A1 fragment activated G protein activated adenylate cyclase to produce cyclic AMP. So high levels of cyclic AMP will be produced, and it result in cystic fibrosis transmembrane conductance regulator. So what happens is that due to this CFTR, that is cystic fibrosis transmembrane conductance regulator, what happens is that this high level of cyclic AMP activate this CFTR. So transmembrane conductance regulator get activated, and so many ions and water from this intestinal cell get into the lumen of the intestine. So water and electrolyte get accumulated into the intestinal lumen from these intestinal cells. So it will lead to watery diarrhea. So 10 to 12 liters of water will get excreted per day through the fecal material of this infected patients, and as a result lot of ions are also get wasted, and due to the lowering of bicarbonate ions, pH of the blood also falls. So this is the effect of cholera toxin. Botulinum toxin. Clostridium botulinum is an anaerobic rod-shaped spore-forming bacteria. It produces botulinum, a protein with characteristic neurotoxicity. Botulism, a severe form of food poisoning, results when the toxin containing food are ingested. Clostridium botulinum is a rod-shaped spore-forming bacteria. It is an anaerobic bacteria, and it produces a toxin called botulinum, and it leads to a food poisoning called botulism. And Clostridium botulinum is common, only seen in canned food items, and when contaminated canned food containing this botulinum toxin is being ingested by individuals, then botulism, the food poisoning disease, which is a neurotoxic disease, can occur. Mechanism of action of botulinum toxin. In the normal functioning of voluntary muscle, a nerve impulse sent along the axon of the motor neuron reaches the neuromuscular junction. The secretion of acetylcholine by the end terminal of the axon then initiate contraction of the muscle. Botulinum toxins bind into the axon near the neuromuscular junction and prevent the secretion of acetylcholine. Muscles cannot contract. If this paralysis extends to the muscle of the chest and diaphragm, death by respiratory failure can result. So, in normal cases, in the functioning of a voluntary muscle, a nerve impulse sent along the axon of the motor neuron reaches the neuromuscular junction, and it will result in the secretion of acetylcholine at the end terminal of this axon, which initiate the contraction of that particular muscle. And the botulinum toxin, the presence of that toxin, it bind into the axon near the neuromuscular junction, which causes the prevention of secretion of acetylcholine. So, acetylcholine is responsible for the contraction of the muscle. So, when this botulinum toxin is present, it will prevent the secretion of acetylcholine. So, muscle contraction does not take place. And if this paralysis extends to the muscle of the chest and diaphragm, so chest muscle as well as that is ribs and diaphragm cannot contract. So what happens is that the inspiratory muscles cannot work properly, and whole process of respiration get affected, and death can occur. So if the paralysis extends to the muscles of the chest and the diaphragm, death by respiratory failure can result. So botulinum toxin, if it is present in larger quantity, then it will cause a systemic infection, and that can lead to respiratory failure and death. So that is all for today's topic. Thank you.